Team number 18, RNA viruses. Plan of the team: structure of RNA viruses, classification of RNA viruses, and properties of influenza virus, parainfluenza, and mumps, measles, rubella, picornaviruses, rhabdovirus, and epidemiology of influenza, parainfluenza, mumps, measles, and rubella, poliomyelitis, and aseptic encephalitis, and pathogenesis of this of influenza, parainfluenza, mumps, measles, rubella, poliomyelitis, and encephalitis, clinical manifestation of these diseases, and laboratory methods of diagnosis. RNA viruses. These RNA viruses icosahedral and helical type, and icosahedral are divided into two, naked and envelope, and helical is divided envelope. And RNA viruses, orthomyxoviruses, paramyxoviruses, and rhabdoviruses, and rhabdoviruses, and enteroviruses, and other types: rhabdovirus, buyanovirus, and arenavirus, and reoviruses, togaviruses, and flaviviruses. Structure of viruses. Viruses contain core nucleic acid, protein coat or capsid, composed of a large number of subunits, rule of protection, envelope or no knot. Virion size range is from 10 to 400 nanometer in diameter. Envelope virus, spike, capsid, nucleic acid present, and naked virus, capsid and nucleic acid present. And the diversity of viral genomes, the nature of the nucleic acid, DNA, and RNA, the structure single-stranded and double-stranded, the shape linear, circular, and segmented, non-segmented. The capsids, two types of symmetry: protective coat made of repeating subunits of proteins, and the helical rod-shaped structures of plant viruses and bacteriophages, and the nucleocapsids of envelope viruses, and icosahedral spherical viruses. Two types of viruses: membrane nude viruses and envelope viruses. Have a membrane derived from the host cell membrane, but modified by insertion in viral proteins, glycoproteins. For example, influenza virus. Virus consists of three genera: influenza A, B, and C. Morphology of influenza virus. Spherical. Influenza virus are spherical in shape, 120 nanometers in size, and helical symmetry. It comprises of a helical nucleocapsid 9 nanometers and surrounded by an envelope. Viral RNA comprises of multiple segments of negative senses as standard stranded RNA. Each segment codes for specific viral proteins having a specific function. Influenza A and B contain 8 segments of RNA. Influenza C contains 7 segments of RNA. The segment coding for neuraminidase is absent. Site of replication. RNA replication occurs typically in the nucleus, in contrast to most other RNA viruses which replicate in the cytoplasm. Viral proteins. Influenza virus contains 8 structural proteins: BP1, BP2, PA, NB, and HA, NA, M1, and M2, and 2 non-structural proteins, NS1 and NS2. BP1, BP2, and PA are the polymerase proteins responsible for RNA transcription and replication. Nucleoprotein is the major capsid protein associated with viral RNA to form a ribonucleoprotein, or nucleocapsid with a helical symmetry. Viral cycle: absorption and attachment to cell membrane via receptors, penetration and uncoating. Next process: biosynthesis of viral RNA and proteins, and next is biosynthesis of viral RNA and proteins. Next, maturation, nucleic acid and viral proteins assembly, and the last process: release of virons by budding. Viral cycle: absorption and attachment to cell membrane via specific receptors. The nature of the receptor is responsible of the viral tropism. The receptor can be a protein, a sugar or complex lipid. Penetration and uncoating, envelope and the capsid are removed and viral nucleic acid is released into the cell. Replication of RNA viruses are quite complex. Single-stranded RNA viruses contain either a positive sense or negative sense RNA. By convention, a positive sense RNA serves as messenger RNA, if positive sense viral RNA serves as M matrix RNA for reproduction of viral proteins. If negative sense, no viral proteins can be made until viral matrix RNA is available, then need to make a positive strand. By a viral RNA enzyme, then the different strategies of replication of RNA viruses result in the synthesis of viral messenger RNA. Positive strand RNA viruses, in this virus, the genomic RNA is the same sense as mRNA and so functions as mRNA. This mRNA is translated immediately upon infection of the host cell. The genomic RNA is translated by the cellular machinery for translation of viral proteins. And synthesis of one and large polyprotein precursor of the structural and non-structural by proteolytic cleavage. Examples: prototype number, poliovirus, picornaviridae, human rhinovirus, picornaviridae, and hepatitis C, flaviviridae. And prototype number, poliovirus. About poliovirus: picornaviruses belong to the family Picornaviridae, which include two major groups of human pathogens, enteroviruses and rhinoviruses. Enteroviruses, they are transmitted by fecal-oral route, and through they multiply in the intestine. They don't cause any intestinal manifestations. They are associated with various systematic manifestations, including poliomyelitis, a childhood flaccid paralysis, which is in the verge of eradication globally. Enteroviruses comprised of 96 human serotypes, poliovirus 3 serotypes, and the Coxsackie virus A 24 serotypes, and the Coxsackie B 6 serotypes, and the ecovirus 33 serotypes, and the enteroviruses 72 serotypes. Serotype 72 is reclassified as hepatitis A virus. And rhinoviruses, they are transmitted by respiratory route and cause common cold. Polioviruses, antigenic types, type 1, it is the most common serotype to cause epidemics of poliomyelitis. This serotype is responsible for all the poliomyelitis. This serotype is responsible for all the natural cases of poliomyelitis occurring globally, globally at present. Type 2, it is the most antigenic serotype, and hence is the easiest serotype to be eradicated. And the morphology of picornaviruses are simple in structure, very small, 30 nanometer size, and non-enveloped. They are spherical shaped and have icosahedral symmetry, and capsid is composed of 60 subunits, each consisting of 4 viral proteins, except paracoviruses have 3 proteins and possess single stranded positive sense liner RNA. Resistant enteroviruses are stable at acidic pH, whereas rhinoviruses are acid label. And heat label all enteroviruses, and some rhinoviruses are inactivated by heating at 55 degrees Celsius for 30 minutes, which can be prevented by magnesium chloride. The poliovirus, the genomic RNA has 2 characteristics. The third end is polyadenylated. The fifth end is not capped but is covalently associated to a small protein of 22AA VPG, and it forms a secondary structure called ERES, and the poliovirus has an RNA-dependent RNA polymerase. And pathogenesis of poliovirus disease. Transmission: polioviruses are transmitted by fecal-oral route, most common, followed by respiratory droplets via inhalation, or rarely by conjunctival contact, and multiply locally. It multiplies in intestinal epithelial cells, submucosal lymphoid tissue, tonsils, and Peyer's patches. And receptor viral entry into the host cell is mediated by binding to CD155 receptors present on the host cell surface. Spread to central nervous system and the spinal cord. Hematogenous spread, most common. Virus spread to the regional lymph nodes and spills over the bloodstream. Primary viremia. After further multiplying in the reticuloendothelial system, the virus enters the bloodstream again and causing secondary viremia. Then it is carried to the spinal cord and brain. And neural spread. Virus may also spread directly through nerves. This occurs especially following tonsillectomy, where the virus may spread via glossopharyngeal nerve present in the tonsillar fossa. And site of infection, site of action. The final target site for poliovirus is the motor nerve ending and anterior horn cell of the spinal cord, which leads to muscle weakness and flaccid paralysis. And neuron degeneration. Virus infected neurons undergo degeneration. Earliest change in neuron is the degeneration of nasal body. Aggregated ribosomes normally found in the cytoplasm of neurons. Pathological changes are always more extensive than the distribution of paralysis. Negative strand RNA viruses. The genomic RNA is negative sense complementary of mRNA and must therefore be copied into the complementary plus sense mRNA before proteins can be made. Thus, besides needing to code for an RNA-dependent RNA polymerase, these viruses also need to package it in the virion so that they can make mRNAs upon infecting the cell. Rabies virus, rhabdovirus. 55,000 persons die of rabies each year. Enveloped virus, genome negative single-stranded RNA and 5 proteins: glycoprotein, nucleoprotein, RNA polymerase, and phosphoprotein matrix and nucleocapsid helical symmetry. The family of Rhabdoviridae comprises of two genera, genus Lissa virus and the genus vesiculovirus. And rabies virus causes a rapidly progressive acute infectious disease of the central nervous system in humans and animals, transmitted from another rabid animal. And also the human cases are few in number. Rabies is still considered as a major public health problem because it is almost always fatal. Morphology. Rhabdoviridae family has a unique morphology, bullet-shaped, 75 nanometer in and 180 nanometer in length, and enveloped. They have a lipid envelope in which 10 nanometer long peplomerase or spikes glycoprotein G are embedded, and envelope is lined internally by a layer of matrix protein. Nucleocapsid has a helical symmetry and comprises a single-stranded negative sense RNA, nucleoprotein, and polymerase proteins. Antigenic properties. Rabies virus has only one serotype. However, there are 7 antigenic variants that are associated with specific animal reservoirs and found in different geographical areas, and the variants have minor differences in their antigens, nucleoprotein and glycoprotein G, as well as in their nucleotide sequences. And the glycoprotein G is a major factor responsible for rabies virus neuroinvasiveness and pathogenicity. It also possesses hemagglutinin activity. Transmission of rabies bite. Rabies virus is usually transmitted to human by the bite of an infected animal. However, other modes of transmission have also been rarely reported. Dog bite is the most common mode, and other animal bites can also transmit rabies. Monkey, sheep, goat, and cat, buffalo, and horse, except rat bite and human bite. Bat bite mostly goes unnoticed. Migrating fruit-eating bats are the most common bats that transmit rabies in America. Human-to-human transmission is theoretically possible, but is extremely rare. Non-bite exposures are rare, such as lick on abrasion or mucosa, inhalation of virus containing areolas and generated from the infected bats, corneal transplantation, and animal susceptibility to rabies virus. Susceptibility varies among various animals. Very highly susceptible animals: fox, jackals, and wolves, and cotton rats. Highly susceptible animals: rabbits, cattle, cats, hamsters, and bats. Moderately susceptible animals: dogs, goats, and sheep, horses, and non-human primates. Rabies virus also undergoes certain changes when it is propagated in animals. Segmented negative strand RNA virus. Besides the order of mononegavirals, there are viruses like Orthomyxaviridae that have many fragments of negative polarity. Their genome are segmented. Example: influenza virus. Their replication strategies are identical. Influenza virus or flu, the cause of flu. Influenza virus. Structure of influenza virus. Enveloped virus genome negative SS segmented RNA, 8 segments present in structure of virus, and and the two glycoproteins, hemagglutinin and neuraminidase. The genome inside of virus, and NS2 antigen present, and M1, M2, and hemagglutinin and neuraminidase antigens present on surface of this virus. Clinical manifestation of influenza virus incubation period, it is about 18 to 72 hours, which directly depends upon the inoculum size and the immune status of the host. Uncomplicated influenza flu syndrome. Majority of individuals are either asymptomatic or develop minor upper respiratory system, such as chills, headache, and dry cough, followed by high-grade fever, myalgia, and anorexia. It is a self-limiting condition and indistinguishable from the infections caused by other upper respiratory tract pathogens. And pathogenesis transmission, it's via infected aerosols generated by cough and sneezes, rarely via contacts or by fomites, and small particle aerosols are more efficient in the transmission. Target cell entry, viral HA attached viral hemagglutination attached to specific sialic acid receptors on the host cell surface that leads to viral entry. Ciliated columnar epithelial cells are most commonly infected, but it may also infect other cells, including alveolar cells, mucous gland cells, and alveolar macrophages. Multiply locally. Virus replicates in the infected cell, and infectious daughter virions and spread to the adjacent cell to involve large number of respiratory epithelial cells over several hours. Spread very rarely. Virus spread to the lower respiratory tract or spills over bloodstream to involve extrapulmonary sites. Double-stranded RNA viruses, example reovirus and rotavirus. Genome with 10 and 11 segments of dsRNA. The virus contains a dsRNA dependent RNA polymerase. The transcription is asymmetric. One of the ds strand is transcribed, and the transcription takes place inside the subviral particle that contains all the activates required for the capping of messenger RNA. Rotavirus are found worldwide, causing major of diarrhea associated hospitalization and 600,000 to 850,000 deaths per year. And Nut virus triple capsid, 11 segments of dsRNA, and 6 structural proteins and 6 non-structural proteins.